So in today's class, we are moving on to chapter 2, and this chapter 2 talks about cell structure and cell division. So in today's class, quite a big chapter. So I've divided into two parts. So we will cover 50% of the topics today, and then 50% tomorrow. So let me give a brief introduction, and then we come back to the PowerPoint slides. So if you take this whole planet or universe, we can divide into two parts: living organisms, and second is non-living. So what are the properties of a living organism? The properties of living organism is, number one, to grow.
Repeat that.
Grow in numbers.
Okay.
Now, this grow in numbers, if you look in terms of cells, it corresponds to cell division. And this process of cell division is very much it's very planned, it is very systematic, and it is also the basis for cellular differentiation in multicellular organisms.
It's also.
And this is also, if cell division doesn't happen in a right way, then it can cause severe diseases such as cancers. So the knowledge of cells is very important in terms of human cells. In bacterial cells, the knowledge of bacteria is important because a bacteria is an infectious organism, right? It can cause diseases. So if we know the cellular aspect of bacteria, then it will help to develop a good antibiotics. And if you look at the plant world, plant kingdom is unique because plants are the primary producers. Right? How they are primary producers? Because they can generate, using sunlight, they can generate food for themselves, right? And we are dependent upon plants. So even bacteria, they want to grow inside the host or wherever it is there. Now, there are some interesting organisms which are between living and non-living, and these are very important for public health, and those are viruses. Viruses, when they are inside the cells, they are growing, they are dividing in number, and their objective is to utilize the resources of the host. But when the virus is released into the environment, it cannot independently divide. It needs a host cell. So viruses most likely were one of the most earliest particles or that were formed when the evolution occurred at a molecular level. So possibly there was formation of water. Water helped in various reaction. Water serves as a various as a medium for various reactions for formation of proteins, nucleotides, glucose molecule. These are all speculation, but as a technology develops and there are model systems, then we can exactly, pinpoint how the evolution happened. So viruses are considered to be one of the earliest developed, sequence of DNA that got into life when it entered the cells. So that's a very, bigger question. But viruses. So let me just quickly go into the structure of virus, because virus as it is has become very important these days. And so I thought that, I'll take this opportunity to talk about viruses in this class before I move to human cells. So broadly, you can divide very, very broadly the viruses that exclusively infects bacteria. They are called as bacteriophage. They don't infect human cells, only bacteria, very species specific. Now, if you look at the structure of this bacteriophage, you will find a head, and there is a stem, and there's a tail. So within the head, there is a genetic material. Now, there are various kinds of bacteriophages. It could be single-stranded DNA, it could be double-stranded DNA, circular DNA. So we are not going into the details. So these are bacteriophages which are present in tons in environment, but their host is bacteria. So this head contains genetic material, and this head is encapsulated in a protein called as capsid. Now, this head is connected to the tail through a cylindrical shape structure called as sheath. And tail is utilized by the bacteriophage to attack a bacteria. And when this bacteriophage attacks a bacteria, or let's say goes and first binds to the bacterial host, then they punch a hole in the bacterial cell wall and membrane, and the DNA or genetic material is injected that goes through the sheath into the host. And this DNA or genetic material tries its best to take advantage of bacterial machineries. So this particular genetic material is responsible for making tons of bacteriophage. Now when the bacterial body is full of this bacteriophage, and bacteriophage is totally dominating over the bacterial system, bacteria will die. So that's bacteriophage. In humans, it is very complex. Some viruses, they have DNA as their genetic material. Some viruses have RNA as their genetic material. And then more complex is the covering that encapsulates the genetic material. This could be called as viral envelope. And within this envelope, there are various apertures that protrude out. These structures, they are mainly protein or glycoproteins. These protruding structures are those objects through which virus attaches to the cell. So virus genetic material, which is present inside, this is also covered by capsid, and above capsid there are envelope, and within envelopes there are proteins, various kinds of proteins, depends on what type of virus it is. And there are some proteins which serves as a receptor that binds to human cells. For example, in coronavirus, there is spike protein. This spike protein has tremendous capacity to bind to the lungs, the cells in the lungs, and then this virus can divide in the lungs. It prefers lungs, although now people are trying to getting cases where the data shows that viruses, people who are diabetic or having heart disease, the virus is present in lungs, but it affects various organs. It could affect brain. Many people during COVID got stroke. So we still don't know much about viruses. We are beginning to know because viruses keeps evolving very fast under the immune pressure. So spike protein, what I want to highlight is the protein through which coronavirus or COVID-19, it binds to the lung cells. And this is the protein against which mRNA vaccine has been developed. And that is the vaccine that we got. So knowledge of this viral structure is very important to develop new therapeutics. There are many companies that are working on strategies which are beyond vaccine, such as medicines and so on and so forth. So viruses, why I brought this? Viruses are the objects which is in between living and non-living. Once it is inside the host, it is dramatically active. Inside in the cells and outside the cells, it is very non-existent. But outside the cells, in the air or atmosphere, it tries to look for a host, right? So that is one of the mechanisms by which through air that many of us got COVID-19. So that's the link between living and non-living organisms. So, right. Now, so we covered viruses and bacteriophages. Now the organisms, specifically focusing on the living organism, right? So what is biology? Biology is the study of living organisms or the study of life, right? Biology. So, but if you look at, wherever you are in New York, if you look at, you will find very different, geographical landscape, and so, and if you look at the organisms, right, there are so many diversity in organisms which are present on this planet. Now, as a biologist, you want to develop an approach so that these diverse organisms can be classified. But it is very difficult, given the degree of diversity, it is very difficult to give one hat to everyone. So you look for patterns which are similar and patterns which are not similar. And then once you understand the pattern, then you try to derive the information, try to understand, So that's why, I gave this exercise in the beginning so that, you know what you know, and then as we advance in this class, you try to derive the pattern in these diverse biological organisms, and hopefully that will help you. So living organism is a very big canvas. So there are various ways in which scientists, they have tried to classify these living organisms, right? So one way could be is by the number of cells. And these could be like unicellular and multicellular organisms, just on the basis. What could be the example? Unicellular could be a bacteria, an algae, or fungus, or fungi. And multicellular could be plant cells, human cells, or any animals or mammals. Then the second way in which you can organize this diverse biological organisms is based upon the presence or absence of chloroplast. Chloroplast is that special organelle that is present only in plant cells. And these chloroplast contains chlorophyll, which is a pigment that has ability to grab sunlight, and then in presence of carbon dioxide and water, it synthesizes food, which is carbohydrate, glucose. So on the basis of the presence of chloroplast, there could be a plant kingdom and animal kingdom. On the basis of number of cells, unicellular and multicellular organisms. The third basis could be by the presence and absence of nucleus. If the nucleus is absent, then it is called as prokaryotes. If it is, nucleus is absent. If the nucleus is present, then those are called as eukaryotes. Okay? So in this class, we will first. So what are the examples of prokaryotes? The example of prokaryote is a bacterial cell, bacteria, and then an example of eukaryotes could be a human cell or animal cell, as well as plant cells. So we will first study about the bacteria, and then we go into human versus plant cells and try to understand how this cell works because cells are the fundamental unit of life. Cells form tissues, tissues form organs, and organs form organ system. So there is a hierarchy in a multicellular organism. There's a hierarchy that there is a cell. Many identical cells come together to make tissues. Several tissues, they combine to form an organ, and organs form a system. And there are many systems in our body, such as respiratory, or let's say circulatory. There are many systems in our body, and these functioning of these systems are important for survival of any multicellular organisms, because we are not dependent upon one organ to function. We are dependent upon many organs to function together. So in our body, many things happen in parallel, and the basis for that is cells, right? So let's go back and start from bacterial cells. So one of the advantages that that we have while teaching synchronous class or virtual class is that you're working with internet, and you can directly go and get some very good pictures, and so that is I believe is very helpful for me to explain, and I feel that students also have. Sorry. Yeah. So this is a typical. So all these figures are also on my PowerPoint, but just looking at the very good pictures. So I hope we all will have a better sense. So this is a typical bacterial cell. So first, let's consider what bacteria wants, right? So bacteria is a unicellular, single cell organism. So it wants the things to be very simple. It has a simple single cell structure. So it wants to be in a diverse environment. It wants to survive in water. It wants to survive over the, let's say, bread or milk. It also wants to float with air. So it wants to be in the water. It wants to be in the air. It can swim inside, milk that is expired. It could hang on to a piece of bread. So that's because bacteria is very sensitive to nutrition. If there is plenty of nutrition around, it will thrive. If bacteria has less food, then it will try to store its energy. It might go into hibernation. It might form some capsule. It depends what kind of bacteria. But here we are taking a very general aspect. So in this bacteria, so bacteria could be of different shape. Here in this figure, bacteria is capsule shape or a cylindrical shape. Now, what you see on the surface of bacterial cell, you see some appendages, right? There is one long appendages. There is one long appendage which is whip-shaped. This is called as flagella. This flagella is mainly used by the bacteria for movement, locomotion. So let's say, so why would bacteria want to locate or move from one place to another? So let's say if there is an aquatic bacteria, then bacteria would like first to move in the direction where food is available. So in search of food, bacteria, flagella will help the bacteria to swim. Also, if there is a threat to its life, then it will try to go up. For example, some bacteria may not like light. So the moment they feel light, they would run away from there. So flagella, for nutrition, for protection, it helps the bacteria to move from one place to another. Sometimes bacteria wants to flow in the direction of the current, that would help. So it will use flagella to control its movement. The second kind of structure that is present on the bacterial cell wall is hair-like structure. So one is flagella, which is very long, whip-shaped, thin. The second kind of structure that is present in bacteria in the extracellular space or on the cell wall is small hair-like structure. There are small hair-like structure that are present on the surface of bacterial cell wall. Right? So all over the bacterial cell, these hair-like structure are present. These hair-like structure is called as fimbriae. This fimbriae helps the bacteria to attach to a solid substrate. That solid substrate could be a rock in a river. That substrate could be a piece of bread. Right? So one is flagella that helps in movement or locomotion. Second is fimbriae There is third type of structure which is present in bacteria. That is also a hair-like structure. That is also a hair-like structure. These structures are called as pili. Now there are no male and female bacteria. There's no distinction of bacteria on the basis of sex, but it has been established that there are a small population of bacteria that contains pili, and they are referred as male bacteria. And through pili, this bacteria can conjugate with a bacteria which could be a female bacteria. And then through this pili, it can transfer its DNA. So there could be a bacteria which has a pili on the surface, and this bacteria can conjugate with a bacteria that doesn't have a pili, and through this pili, the bacteria can conjugate and can transfer its DNA. So hypothetically, it becomes a male bacteria, and this becomes, but there's no well-formed sexual organ that one can distinguish between a male and a female bacteria. So we discussed those apertures that are present on the surface of a bacterial cell. Okay. Now let's look at the bacterial cell wall and cell membrane. So bacteria, it lives in an open environment, right? It doesn't have layers of tissues like our body. So our body has outer skin cells, which are mostly dead cells. Then we have epidermis, then dermis. So there are layers of protection in our body. In bacteria, it is an open atmosphere. So bacteria has a very strong covering across its cell, right? So bacteria is intracellular. So they have a very strong covering. This is called as cell wall. This cell wall is mainly dead, and it is permeable to everything. Mainly, it serves as a border. It tries to protect the bacteria in adverse conditions like excess heat or excess cold or change of pH. So there is cell wall which is dead and it is permeable. But very interestingly, there is another layer of membrane that within the cell wall that is called as cell membrane. Right? Within the cell wall, there is another covering or another coat that is called as cell membrane. Now, the difference between cell wall and cell membrane is that cell membrane is a living and it is semi-permeable, meaning that so cell wall could be free for all. Cell membrane is semi-permeable, means that something can go inside, something cannot go inside, right? So bacteria has three apertures or three, types of extracellular like fimbriae, flagella, and pili, and it has two covering. One is outermost is cell wall, which is dead and permeable, whereas cell membrane is living and selectively permeable. Now, as I mentioned to you that bacteria, the basis of bacteria is it's a typical representative of prokaryotic world, and the basis is that it doesn't have a nucleus. So what it has, of course, bacteria has DNA, but this DNA of bacteria is not present inside the nucleus. So when we make progress and we go to mitosis and meiosis, you will see that human chromosomes or animal chromosomes are very organized, and they're also again protected in a separate organelle called as nucleus. But in bacteria, the DNA, it is present inside the cytoplasm. There is no boundary between the DNA and cytoplasm. So this bacterial DNA is diffuse. It is not, so remember this, the structure of bacterial DNA or chromosome, this is diffuse and present all across, diffused in a bacterial body. And this is called as nucleoid. So nucleoid is also referred as false nuclei, but the important message is that there is no nucleus. The chromosome DNA of bacteria is diffused and it is present in the cytoplasm. So what is cytoplasm? Cytoplasm is a pool of fluid inside bacterial cell, which contains various proteins, inorganic and organic compounds that would help bacterial physiology. And as I mentioned to you, one of the important component of cytoplasm, it's fluid in nature, and one of the important component is water. Some bacteria also have capsule, right? So these bacteria are very complex. It's if they are infectious, then it is very difficult to control their growth inside human body because they are capsulated. They have an extra layer in addition to cell membrane and cell wall, and they provide thick covering to bacteria. So for bacterial interest, it is protection, but for host, it is disadvantage because it prevents the antibacterial treatment. Ribosomes, so one of the most important organelle. So a cell is a factory which is supported by various organelles. Now, bacterial cell is simplest cell. They have DNA, cytoplasm, various membranes, cell membrane, cell wall, capsule. Ribosomes. Ribosomes are those organelles which help the bacteria to synthesize protein. So it's a very simple structure, right, of bacteria, bacterial cell. So let's take 5 minutes break, and then we come back. So I see few more students than yesterday. You can just tell me your name. I have taken attendance in the beginning of the class. So one is Brianna Laguerre, I got you. And then Sane Bloomfield. Anybody else?
Sania Kelly.
So
Barry.
Sania Kelly.
yeah. So those who are attending the class for the first time, you have an introduction on the blackboard. And that is the first activity that I gave to the class. And all these activities that we have in the class, it carries points. So I'm just repeating myself.
Oh, I did it already.
Okay, great. Do you have any questions?
Professor, I
No.
was about the handwriting part of the assignment. Is that
Yes.
mandatory?
Yes. I mean, you will lose some points, but it is yes.
Okay, thank you.
Was supposed to take effect. The TLC originally agreed.
Professor, I wanted to make sure if you got my attendance because I was late today.
May I tell you, could you tell me your name?
Daniel Forbes.
Yeah, I got you. Thank you.
My name is Sania Kelly.
Who? Yeah, I got you, Sanya.
Okay, just making sure.
Yeah, no problem. So I was, the first activity that I gave, early in the morning, and a few of you got very right. And this is a very, clean way diagram to, to present what are the differences or similarities between a plant cell and an animal cell. So the biggest physiological difference is that plants are very independent. They are not dependent. But animals, humans including, we are dependent upon for source of energy. We don't have ability to, to directly convert solar energy into carbohydrate or glucose, which as I mentioned in the last class, glucose is the major source of immediate energy. So plants, now here you can also draw some similarities between bacteria and plant cells. Plant cells also, they are open in the environment, so they have cell wall, right? And they are very tough and but permeable. So that's one of the similarity between plant cells and the bacterial cells. Now coming back to the plant and animal cells and animal cells both have ribosomes. Ribosomes are the major organelle that are responsible for the synthesis of protein. And protein, as we talked about in chapter number 1, is a major source for defense, for our body, enzymes, hormones, infrastructure. So all these proteins are synthesized by ribosome, on the surface of ribosome. Then, what is another organelle that is common between plant and animal cell is endoplasmic reticulum. This endoplasmic reticulum is actually two types. One is a smooth endoplasmic reticulum and one is rough endoplasmic reticulum. The smooth endoplasmic reticulum is mainly involved in lipid biosynthesis. The rough endoplasmic reticulum, it is rough because on the surface of this endoplasmic reticulum, there are small ribosomes. So it makes them appear rough. And this rough endoplasmic reticulum is also involved in the synthesis and processing of proteins, which have different functions. They might be involved in trafficking, meaning that these proteins have to be secreted out of the cells. So these are processed inside the rough endoplasmic reticulum. Plasma membrane and cell membrane, they are same terminology. Some books will not call some books will call cytoplasm as cytosol or plasma. Some books will say cytoplasm and will say plasma membrane, but plasma membrane and cell membrane, they are the same things. Golgi apparatus, it helps in protein modifications and secretions. So Golgi apparatus and endoplasmic reticulums, they work with proteins, but the primary organelle that helps in the synthesis of protein is ribosome. Endoplasmic reticulum only handles those proteins that has to have some modifications so that the protein can be easily secreted out of the cells. Golgi apparatus also helps in protein processing so that these proteins could either be glycosylated, lipid moiety could be attached to the protein, sulfate moiety could be attached to the protein. So all these proteins processing happens in Golgi apparatus. Mitochondria is that organelle. I'll come back to it when I come back to my PowerPoints. Mitochondria is that organelle which is called as powerhouse. Yesterday, we talked a lot about ATP as a currency for energy in cells. This is generated inside the mitochondria. So mitochondria is the energy production organelle. And then the nucleus. So both plant and animal cells, they are eukaryotes. They contain nucleosomes. Sorry, they contain nucleus, and within this nucleus is present DNA. But DNA is not diffused as bacteria. DNA inside the nucleus is packaged into chromosomes. Right? Now, these are the similarities between plant cell and animal cells. What are the differences? The difference is vacuole. There is a large vacuole in the plant cells. Now you can ask me a question, why does plant cell contain vacuole? So plant cells, they are in open climate, open environment, and they're directly under the sun. So these vacuoles, they contain water and air that helps to protect plant from dehydration. They maintain the turgidity of the leaf. So chloroplast, not every part of the plant contains chloroplast and chlorophyll. It's only the leaf, the green part of the plant. And the leaves are very, very soft, part of the plant. And they contain vacuole because leaves are extremely sensitive to heat. So these vacuoles, helps the plant to stay hydrated. They maintain the osmotic pressure, so on and so forth. Whereas in animal cells, there are small vacuoles. Now, vacuole in animal cells become important, especially in single cells such as algae or fungus. They also help in maintaining the turgidity pressure, osmotic pressure, because they're directly under the environment, which could be harsh. But there is a second kind of vacuole also present in animal cell, which is called as lysosomes. These lysosomes contains enzyme. So in humans, in our body, we have a well-formed digestive system where we can ingest the food, and food is digested by moving down to stomach, small intestine, large intestine, and the waste is thrown out. In single cell eukaryotes, there is not a sign of digestive system. So these are small vacuoles called as lysosome, which contains digestive enzyme, which digest the food that is captured by this unicellular cells, which could be an algae, for example. Centrioles and centrosomes, they help the animal cells to during mitosis for chromosomal movement that we will see. Some of the animal cells, especially the single cell eukaryote, they contain flagella. So look at algae. Algae also coexist with bacteria in water, and they need to move. So small single cell eukaryotes such as blue-green algae, such as Chlamydomonas, they need to move in search of food, to protection. So, and as I mentioned to you, chloroplast, it contains chlorophyll, and this chlorophyll helps in capturing the solar energy, and this chloroplast is absent in animal cells. There are exceptions. Single cell eukaryotes, such as blue-green algae or Chlamydomonas, they contain chloroplast. So these algae, these algae are supposed to be in the course of evolution, a transition between plant cell and the animal cells. So one figure I wanted to show you, the nucleus, right? So nucleus is present inside the cytoplasm. Nucleus is separated from the cytoplasm through a membrane, through a boundary that is called as nuclear membrane. Some books will refer this as nuclear envelope also. And within this nuclear envelope, there is also a small spherical shape organelle called as nucleolus. They play an important role in RNA biology. And in contrast to to the bacterial DNA, which is diffused. Animal cell chromosomes, DNA is present as chromosomes, which are very organized, very organized structure. Humans have 23 pairs of chromosomes or 46 chromosomes. Now, in this figure is not 100% accurate, because in the nucleus, particularly in the nuclear envelope or nuclear membrane, there are some pores. Through this pore, the nucleus communicates with cytoplasm, right? So it's not like a solid membrane, right? It has a nuclear pore through which various nuclear protein and mRNA or RNA, rRNA or tRNA, they are exported out of the nucleus into the cytoplasm. And there are some proteins which are imported from cytoplasm to the nucleus. So this is not a very, so one has to be very careful, when you're teaching, or especially teaching, a lot of responsibility when you're. So when you go and look at things, what is there on internet, you're surprised that some of them are so good, but they are small mistakes. So that's why I chose this figure. So hopefully you'll try to remember that. So nucleus has chromosomes, it has nucleolus, and it has nuclear pores through which it communicates. So let's go back to the PowerPoint. So we talked about bacterial structure. This figure is from the book. This is the picture of eukaryotic cell. Here you can appreciate the difference between smooth and rough endoplasmic reticulum. Right. So you have a smooth endoplasmic reticulum that participates in lipid biosynthesis. Then there is rough endoplasmic reticulum, which are called rough because they have ribosomes, which helps in protein, special kind of protein synthesis and transport, especially those protein that has to be secreted out. Then you also have lysosome, which contains digestive enzymes, centrioles also. So you need to know the function of each of these organelles, right? Then there are cytoskeletons, which gives, so so our body stands against gravity, and one of the ways in which our body is able to stand against gravity is through development of vertebral column, the backbone, right? So we can stand against the gravity, and that is supported by bones and muscles, right? For us to do the work. And so inside the plants, there are also infrastructure like muscles and bones. These are called as cytoskeleton, right? So bones are cytoskeleton, right? Bones are the major element of skeleton, right? And so cytoskeletons are microtubules, intermediate filament, microfilament. So microtubules or spindle fibers we will study while we study mitosis and meiosis. Now let's go to the next topic, which is called as lipid bilayer of plasma membrane. At this point of time, do you have any questions with regard to the topics that I have covered? All right. So as I mentioned to you, that plasma membrane is the first covering beyond cytoplasm. In bacteria, in a typical bacterial cells, from going from outside to inside, there can be capsular bacteria, there is cell wall, and then there is plasma membrane. Plasma membrane is a living organelle structure that is semi-permeable. And it is present in plant cells, animal cells, and bacterial cells. Animal cells may not have cell wall and capsule, but plasma membrane is very important part. It is semi-permeable. So now I mentioned to you in yesterday's class that a major component of plasma membrane is lipids, particularly phospholipids. So given that such an important role that provides selective permeability to our cell, scientists wanted to look at the structure. So first of all, a plasma membrane is not soluble in water. It is water insoluble. But it has some property to dissolve in water that we will look into. So plasma membrane is just like if you put a drop of water, a drop of oil in a glass, you will see that that oil forms a droplet. If you add water over water, you cannot know how can you distinguish water from water? But if you add just a tiny drop of oil in a glass of water that you can see from outside, you will see that these oils they form small spherical-like structure. Now, why and how they form? Let's look into the biochemistry part of it. So one thing is given that cell membrane is made up of phospholipid. But how does these phospholipid arrange themselves in a cell membrane? So let's look into. So a lipid molecule consists of a globular structure. This globular structure is called as head. And they also contain thin tail. This is, for sake of understanding, this is tail. Now, it was found out that the head has charge. It could be a mixed charge of positive and negative. Head. Because of this charge, the head is called as polar. Now, this tail region, there is no charge. This is called as non-polar. So head polar, this is non-polar. Now, because it is charged, this can interact with water. Remember the structure of water? Water has an angular structure, right? In which hydrogen is positive and oxygen is negative. So this polar head can interact with water. Hence this, and they like water. So this is called as hydrophilic. head is called a hydrophilic. So positive can interact with negative oxygen, negative can interact with hydrogen. And this non-polar tail do not like water. So they are called as hydrophobic. Now, don't you think that this makes a very unique molecule? That in one molecule, there are two different opposite characteristics. So within one molecule, there is, you see hydrophilicity, and you also see hydrophobicity. So such molecule which have, so what for the sake of understanding, these properties are 180-degree opposite, right? Liking for water, disliking for water, right? So within a single molecule, you are seeing two biochemical characteristics. Such molecules are called as dual characteristic molecule based upon its liking for solvent is called as antipathic molecule. So a lipid molecule, which is the major component of water, is called as amphipathic. Now, let me just, for sake of an example, frogs, right? They're called as amphibians. They have dual characteristic. They can live on land. They can also live inside water. They can be on land, terrestrial. They can be aquatic. So they can survive in both the worlds. So this is called as amphi, means dual. So it can survive in two different habitats. So here, on the basis of biochemistry, this is a lipid molecule is amphipathic molecule because it has both component, hydrophobic and hydrophilic. Now, how these lipid molecules are arranged in a membrane. So these lipid molecules, they are arranged. So let's say, for sake of understanding, that this is the plasma membrane. This is the cytoplasm. Here it is nucleus. And within the plasma membrane, the lipid molecule is present in two layers. Two layers. Now, what you should note here, that head is either pointing outside or inside into the cytoplasm, head. And the tails are facing each other. Right? So tails are facing each other inside, and head is either pointing outside or inside cytoplasm. So a membrane, plasma membrane, which is semi-permeable, is made up of bilipid layer. And while this, both heads, they help in movement across. There are many things need to move inside the cell. For example, water, sodium, potassium, many other cofactors. So these heads and tail, they combine their properties to make plasma membrane as a semi-permeable structure. Okay. This is what you see in this slide, that you have non-polar tail pointing inside, facing each other, and heads are exterior and interior. Okay. So I mentioned to you that lipids are very complex molecule. They are not as simple as protein, carbohydrates, or nucleic acid. So lipids have two components, fatty acids and glycerides, and saturated fatty acid and non-saturated fatty acids. I don't want to go into the details of chemistry, but saturated fatty acids are derived from plants, and saturated fatty acids are derived from animal food, whereas unsaturated from plant products. Right? Glycerides could be neutral or phosphoglycerides, and then you have this steroids, sphingol. So there are huge family of lipids as compared to proteins and carbohydrate. Now let's look at the structure of mitochondria. Why mitochondria is important? Because mitochondria is a place where ATP is manufactured. So this is a powerhouse. But what is the most interesting aspect of mitochondria? And it is the subject of recent research. First of all, hopefully you might find this interesting, or maybe you already know, that we inherit chromosomal DNA from our parents, both the parents. Chromosomal DNA means the DNA chromosomes that are present in the nucleus, half from father, half from mother, father and mother, both. But mitochondria, we always get from mother. That's what current research tells. In future, it might change. But mitochondria, we get from our moms. And mitochondria is the powerhouse of energy. Another most important aspect of mitochondria. So why I said this is the topic of current research, because most of the research focused on the chromosomal DNA. And mitochondria, we were satisfied to know that, okay, this is a powerhouse for ATP generation. But when scientists looked inside the mitochondria, they found that mitochondria has its own DNA. Right? It has its own DNA. It has its own ribosome, which makes it makes people to think that at one point of evolution, mitochondria existed as an independent organism. It might be a special kind of bacteria that has ability to synthesize ATP on its own. And during the course of evolution, there might have been understanding between two different cells in which these two mitochondria and the cells, they would have made a compromise to live together, symbiosis. Because what else could be the sense that DNA and ribosomes are present in mitochondria? So mitochondrial DNA can also make protein. Ribosomes are there to help make protein, right? So these are the subject of intense research. But what does exactly mitochondrial DNA do? What kind of protein is made by the ribosome inside the mitochondria? And now people are learning that there are many diseases also associated with mitochondria. So a mitochondria is, number one, we get from our mother. Second, it has DNA that makes us to think that at a given point of evolution, it might be an independent organism, let's say bacteria, which can synthesize ATP. Now, if you look at, so bacteria has inner and outer membrane, right, which is analogous to cell wall and cell membrane. But inside the body of mitochondria, you will find these folded structure. These folded structure is called as cristae. Right? These folded structures, various knobs, and you'll find fold. This is called as cristae. And this cristae, this folded structure, is present in the matrix, which is analogous to cytoplasm or cytosol. So on this cristae, there are various proteins which are located that helps in the synthesis of ATP. So that's about mitochondria. Then you have lysosomes, right? Lysosomes are those small organelle which contains digestive enzymes, especially in unicellular eukaryotes. So there can be two kinds of vacuoles in animal cells. One that maintains the osmotic pressure, hydration level, and the second is lysosome that keeps the digestive enzymes for digestion, and after digestion the waste is thrown out through these vacuoles. In plant cells, the vacuoles are large size because they have to stand the temperature of the outside climate. So now here we are going to change gear and go into DNA replication. So let's take a 10 minutes break. And in the meanwhile, if you have any question, then please put it in the chat box. So it is 10:37 now. We take break till 10:45. Thanks. So I during the break, I was preparing this Padlet. So I want you to answer so open this link, click this link. And again, I want you to go back and answer the same question. What are the differences between plant and animal cells? So I can understand how much you can recall or how much you're retaining.
Hey, Professor, I'm having problem with this answering the question, just finding where to do that.
Say again?
I say I'm having problems just finding where to put in an answer to your question.
Do you see the screens these students are putting in? You just have to
I'm
press
seeing
them.
that.
Say again?
Yeah, I'm seeing that.
So
I'm seeing
can anyone explain this, how they are doing, so that maybe a student can explain better than me?
Click the click the plus sign at the bottom of the screen when you open it, and then you just start typing.
Oh, okay, so when all I see is a little black little screen. I don't see
Yes,
where
there
to
is a plus. There's a plus sign at the bottom, at the very bottom of the screen. Click it.
right and
Right,
start
and then
typing
where it
again.
says subject, write your name, and then you type your answer at the on that blank screen,
And publish.
and then publish it to send it.
When you publish,
Yeah,
we all see it.
yeah, but I'm typing on that black little thing, and I'm getting nothing is typing.
Where it says subject, do you see subject?
Yeah,
Write your name. Tap the screen and type your name.
yeah, I'm doing that. I'm not somehow it's not
Are you
typing.
able to share your screen?
Let me allow that. I have to allow that to share the screen. Yes, share your screen, Mr. Tony. Yeah, somebody suggesting to refresh the page.
Share your screen so I could see what the problem is, because okay, go in the Zoom. The Zoom that we're in, right? Click the meeting. Are you having internet problems or something? Hello? Hello?
So, okay, I want my screen back if you end, stop sharing.
I think it was his internet, maybe.
Yeah, I think refreshing the page is also a good idea. So you have 4 more minutes. Have you used this before, class?
I have never. Oh my goodness. I have never used it before.
Oh,
No, this is my first time actually using
yeah, I learned it a couple of months back. So it's a good thing to, it's a good platform. You have, it's like a wall. You can write anything you want.
Mhm.
I attended a workshop, training. Then got this package. So you'll get extra points for doing this because I'm doing it for the first time. So in a way, you are helping me. You all are helping me. So you get some extra points for doing this, for participating. So thank you very much for doing this, like, as an instructor, we also learn a lot. I learn a lot from my students too. Mr. Tony Williams, how are you doing?
I'm making a mess of everything here.
Yeah, take it easy, step by step.
Yeah,
Best
this
is
is
talk
frustrating.
to, I sometimes, let's say I also use Blackboard at one end and you all also use, but it's a little bit different. So it's better that, a student talks to student. So, because I cannot see what you are saying.
I was able to use it on Tuesday, I think.
Yeah, it's very simple.
Um,
I think one of the students mentions that refreshing of page, something like that. What kind of computer?
Yeah, you have to refresh your page because when I was talking to you over the thing and I think you got frozen. So I think you have a connection problem or maybe your keyboard or whatever platform you're
You
using
have
it on.
PC or Mac?
A Mac?
no. Just an HP.
Oh.
It's PC, right? So just refresh it. Yeah.
Yeah, you may have a
And also
connection
try to
problem.
update your computer. Maybe, I don't know.
No, it's nothing like that.
No.
Not like that. It worked on Tuesday.
Two more minutes, please. I think everybody's done. So those who wanted to do. So just anyway, one more minute. So this converts into PDF and I save it on today's date, and then I will, so that, I can give extra, make a list of students who are participating. So, okay. So let's go to the slide. So we are now changing gears, and so we looked at the cells. So we look at let's say we looked at the plasma membrane, its composition. We looked at the cellular differences between bacteria and a human cell, a plant cell and an animal cell, and animal cell, and looked at the functions of various organelles. So now, strategically, we are moving from cytoplasm to inside the nucleus. And inside the nucleus, you have DNA. And DNA, when we covered DNA yesterday, chemical structure of DNA, And now we are going to study the function of DNA. One of the functions of DNA. Right? So what is one of the function of DNA is to keep up. So what is the nature of a cell? That a cell, one cell divides into two, then two cells divide into, 4 cells, and 4 into, 4 into 8 and 8 into 16. So ultimately, the cells grow logarithmically. So if this is the time, if this is the time, and this is the number of cells, then cells grow kinetically, logarithmically in this way. Right? Now, for each cell to divide, right, it won't make sense if the DNA also doesn't divide with the cell division. Because if a cell doesn't have a DNA, then it's a ghost cell. It will die, it will perish. So it's very important that cells, before the cells divide, I repeat, before the cells divide, the DNA should also duplicate so that each daughter cells receives, DNA. So how is that possible? So that process is made possible, this process is made possible by a step called as DNA replication. Now this DNA replication is carried out by an enzyme called as DNA polymerase. So what happens during a DNA replication is that, so let's say this is the, this is the parent DNA. So what will happen when the DNA has to replicate, this DNA strand will separate. So double strand becomes single strand. And then a new daughter strand is synthesized. A new daughter strand is synthesized. I purposefully drew these. So the old parent strand is greenish, right? And the new is like red color. So I purposefully made this kind of diagram is because so this is the parent DNA, and these are the two daughter DNAs. In this daughter DNA, for the formation of, let's say, DNA replication to occur, the parent DNA first separates into single strand, and then after separation, this parent strand serves as a template for the synthesis of new daughter strand. So half the DNA is from parents and half the DNA is newly synthesized. So such mode of replication is called as semi-conservative replication, where one strand is old, one strand is new. But the significance of old strand is that, the importance is that this strand, the old strands, serves as a template for the synthesis of new strand. Now, what do I mean that it serves as a template? Let's take an example. So let's say this is the, so let's take the exact thing. So let's say this is the parent DNA, double-stranded. And you know that these two strands are connected by a by double bond with T, T with double bond with A, G with triple bond with C, and C with triple bond with G. So let me just draw. So this is G with C, triple bond, C with G, triple bond, A and T, T and A, double bond. So when the DNA replication has to occur, these two parent DNA will separate out, right? And what are the things here? This parent strand has A, then T, then G, then C, right? And this parent strand has T, A, G, G, and C, right? So this parent strand is like this. This parent strand is like this. So that now the enzyme DNA polymerase comes in the process.
Professor,
Yes.
correct me if I'm wrong, but if that one is G, shouldn't it be transcribed to C and the last one be G
This
also?
one? This one?
Yes.
Yeah. This should be C. I made a mistake.
Yeah.
Okay. So let me just complete. So now DNA polymerase comes and new DNA strand is synthesized. New DNA strand is synthesized. So this will be let's say C, C, and this will be G. So this is okay. Oh, so mess. So let me just do this again. So the parent strand separates. So this will be here T, A, C, G. So DNA polymerase comes in and synthesizes the new DNA strand. Now, what will be the corresponding base here? It will be T, A, C, G. On this strand, it will be A, T, G, C. See? So now, what you see here is this was the parent DNA, and now you exactly have the two daughter DNA. So this is possible by this enzyme called as DNA polymerase. So let's go back to the PowerPoint. So this is the process of DNA replication that is carried out by DNA polymerase. So there is a parent strand. There's a parent strand that separates, and each strand serves as a template for the synthesis of daughter strand. And what is the rule for the synthesis? The rule for the synthesis is that complementarity, that A pairs with T and G pairs with C, or C pairs with G. So DNA is a chemical molecule. It is made up of carbon, hydrogen, nitrogen, oxygen, phosphorus, but its functional outcome is very biological. Number one, it supports the passage of heredity information from one generation to the second generation. Second, it plays a very important role in cell division. Every cell that has to divide has to undergo DNA replication first. Okay. Now, our cells, human body, human cells, and you look at one cell, it has nucleus. Within the nucleus, there are chromosomes. And if you look at one chromosome, so many books will show this picture of chromosome, which is right. But in actual, this is the chromosome structure of a typical chromosome. So there is a centromere, and this centromere is connected to two chromatids. The short is called as P, the long is called as Q. So there's a centromere, almost central region, connected to two chromatids. This is one chromosome. When DNA replication happens, this is the product. You have now two chromatids here. The relationship between the old and new is sister chromatid. So this is a single chromosome. They duplicate after DNA replication. The relationship is they're exactly same, so they are called as sister chromatids. Now, I'm sharing with you a very interesting point. So humans, we have two sexes, male and female. Females have 46 or 23 pairs of chromosomes. The last pair, the 23rd pair in females is XX. Both are same. The sex chromosome in human females is XX. In males, it is very different. The 23rd chromosome in males is not XX, but XY. What does it suggest? Suggests that human females have 23 different chromosomes. Human males have 24 different chromosomes, 24 in reality, because of X and Y. The sex chromosome in male is X and Y. So human males have 24 different chromosomes. Human females have 23 chromosomes. That's because of the sex chromosome in female is XX, but the sex chromosome in male is XY. And because of that, there are different biological outcomes that people are still studying, right? XY, XX. Now, so first of all, I discussed with you the typical structure of a chromosome. What is the structure of a duplicated chromosome? Then, so 46 chromosomes, right? In male and female. 46 means, so many students get confused. 46 means 46 different chromosomes. No, 23 pairs of chromosomes, right? 23 chromosomes in two sets. One set is coming from father, one set is coming from mother. And males have 24 chromosomes, 24 different chromosomes, and females have 23 chromosomes. So these are some of the facts that I'm sharing with you that I want you to keep in mind when we start cell division tomorrow. Now, before I end this class, I just want to go to the Blackboard page because you will be taking quiz 1 tomorrow. So just want to make sure that you know from your, and I do my best to show you where the quiz 1 is located. So so that there are not, so sometimes I don't check my email. So even if you're writing, hey professor, I can't see the questions or the test, then then I can't do anything, and it's very difficult to explain everything via email. So let me just show you the Blackboard page. So your quiz 1 is located here. Can everyone see here? This is a folder for quiz 1. And if you click this folder, you will see quiz 1. There are 30 multiple choice question on quiz 1. You have to pick the best option. So when you press this, you will get to your end of at this point of time, the configuration of my Blackboard is very different from yours. So this exam is available to you starting 12 midnight, which is 6th January. And the exam is available to you until 11:59 on 6th January, so end of the day. 30 questions. There are you have been given 35 minutes, more than 1 minute per question, and you can take 2 attempts. Now, if your computer doesn't work, there is nothing I can do. So make sure your internet connections, and you all know as good as me how important these electronic gadgets are, especially in virtual settings. So do you have any questions?
No.
Yes, Professor Miss Savage, Stacy Savage. Is an issue with downloading those questions, and
Which question?
nowadays PD files want you to pay them.
Which question?
Unfortunately, I don't have the money to pay the PD
Which
file.
question? Miss, which questions?
Excuse me?
Which questions are you talking about?
I'm trying to explain so know where I'm coming from. The question, the 30 question, what you have on Blackboard,
Yes.
it's hard to download it for me and a lot of other students. And I got it from a student that able to get the question, pull up the question, and the student screenshot it. Could we end up develop a group chat? So that was even better for us that we did create a group chat yesterday, because for the student who's stuck on can't download the questions, we in problems. So I'm asking you, is it possible, since we can't download it or do the assignments on Blackboard, is it possible, can I and other student email the assignment to you? Because it's not our fault.
No. I think it is much simpler that you are thinking. Can you see the question, miss?
No, not at all.
I agree. It keeps having an error whenever you open it. So
when you first click on it, it gives you
Hold
the
on.
but there's an option for you to view or download. So I get the same pop-up. So when you click the link, it says error message, but there's an optional where it says view or download. So if you hit view, you're able to see it in the application that you have or download it.
I did all of that.
What type of files do you guys have that is opening like that?
It's opening in PowerPoint. It's opening in Microsoft Word.
Yeah, I think I have Microsoft Word and it works for me. Just okay, I'm going to put my number in the group chat because I'm able to access it. So if any of you guys should have a question or a problem, you guys can call me and I could maybe be of some assistance. My number is right there.
And he also wants it handwritten. So even if you're able to see it, you're writing out the questions to upload it.
Right, that's what I'm saying.
Right, they're uploading. So regarding the Blackboard issue, I don't know if it's can you guys go in like the discussion part at least? I mean, without opening the file, like, can you guys at least, like, where the area where it says submit, can you guys access that part?
I could access that part, yes, but it's
Yeah.
just that question I cannot.
Okay, so what I will take the responsibility to do is add me to the chat, and I will screenshot all the questions to you guys, and you guys can submit it on Blackboard. I will
The
personally do that.
screenshot is already there with the question. That's what I was explaining to the professor. The thing is that how can I submit it on Blackboard after I do my assignment? Answer the question.
Do you have do you have a cell phone?
I have a cell phone and a laptop, and they both giving me the same issue.
So you just take the picture of your answers by cell phone and then upload it.
Where?
You can email yourself and then upload it also via desktop. Hold on, Professor. I think she's saying, how does she upload it? Go in your gallery. When you take the picture, go in your gallery and save it as a upload it as a PDF. Then what you would do is that go on your whatever laptop or wherever you take the picture on. I suggest you do it on your phone because it will be easier. Just click where it says Dropbox or add a file. You add the file to it, click it, and then submit it.
Okay.
It's just how you would upload any picture.
Right.
Okay.
So you take the picture of it, then you put it in like a PDF. You can even upload it to like your Google Docs or whatever platform that you have. Save it, and then go on your computer and go in your your files. Click that file, attach it, and then just click submit, and you're done.
All right. Thank you.
May I know the name of the student who is helping out? I'm not able to see, and I'm sharing the screen. Is it Christine Hay? Where are you helping? Miss Savage?
Yes.
Yes.
Thank you so much.
No problem.
So, I would like to end the class now.
Yeah.
And see you tomorrow.
Have a good day.
You too. Bye.
Goodbye, Professor. See you
Bye. Have
tomorrow.
a good day. Thank you. See you tomorrow.
You too.